Welcome back for more AP Biology. Today we're going to be dealing with the last part of the chemistry-ish stuff, or the hardcore chemistry-ish stuff, though we're going to do more of it. But we're going to look at fermentation and cell respiration. So we're going to look at how does it compare to photosynthesis, which is what we dealt with last time. They asked questions about this. So last time we looked at how we can make ATP through the use of light and moving electrons around. It turns out there are actually two different ways that we can make ATP, or cell energy. It doesn't have to say ATP. It could be GTP or TTP or CTP, whatever the A, T, C, and G stand for. The two ways turn out to be one called substrate level phosphorylation, which is the picture on the left, which is you take an enzyme and it can literally take ADP and something with a phosphate, and it moves the phosphate from the object, the substrate, and puts it onto ADP to make ATP. We call that substrate level phosphorylation. The other versions of phosphorylation involve a membrane potential. So that would be either photophosphorylation, which was the light-dependent reactions, or what we're going to deal with today, which is called oxidative phosphorylation. Both cases, we move electrons, makes a proton gradient and a charge gradient. We let those move through this slow spinning turnstile that we call ATP synthase, and the result is we make ATP. Both of them turn out to work. When we look at cell respiration, or the basic ways that we obtain energy if you can't do photosynthesis, the most central pathway is referred to as glycolysis. And we call it the central pathway for several reasons. The first one is almost everything that's alive on Earth will do it. Second one is it doesn't require oxygen, which means it probably evolved a long time ago, and we don't need to worry about actual respiration. Third thing is it can occur inside of any cell's cytoplasm. We don't need special parts to pull this trick off. It occurs anywhere. We usually say it starts with glucose, but that doesn't need to be true. It could actually start with basically anything we want. So what do we do during glycolysis? Quite simply, we take glucose and we snap it in half. That's all we do. In the process of snapping it in half, we're going to make two sides that are perfectly equal. Glucose has 6 carbons. We're going to make 2 3-carbon molecules called pyruvate. And in the process, we're going to produce ATP, and we're going to produce this weird thing called NADH, which is an energy carrier. We've seen it before, or a relative of this before, with the light-dependent reactions with the formation of NADPH. Very similar, but there's no phosphate added to it. What is it? It's an electron carrier. That's all it turns out to be. And electrons are energy. So that's all we do in glycolysis. We take glucose and we snap it in half. The catch becomes, what can you do after this? You have lots of ATP. I mean, we can produce lots of it because we have lots of ADPs. What we don't happen to have a lot of is NADH to turn into NADH. So in order to do this, we need to have some type of recycling mechanism. So if I turn glucose into pyruvate, I use up NAD to make NADH. In order to keep that process going, I need to turn my NADH back into NAD. And I need to keep that shuffling going on back and forth. We call that shuffling of electrons redox chemistry, which just means we're moving electrons around. And there turn out to be basically two ways that we can do this. Option number one is I just take my 3-carbon molecule, my pyruvate, and I change its form a little bit, and we call that lactic acid. It's like when you make cheese or yogurt or soy sauce or something like that. The second option is I could take my 3-carbon sugar, my pyruvate, I'll rip off a carbon in the form of carbon dioxide, and I'm left with a 2-carbon compound that we call ethanol, which is either alcoholic drinks, or it's the smell of bread, or what have you. So we have two options to recycle NADH back into NAD. We either turn it into lactic acid, or we turn it into alcohol with CO2. Those are our two options, assuming there's no oxygen. If there is oxygen, another option exists, and that option is what we call officially cell respiration. Glycolysis isn't really part of cell respiration, even though we're usually sloppy with our language and we say it is. Cell respiration actually occurs inside of the mitochondrion. So if I look at the mitochondrion, what do I happen to notice? I see two sets of membranes, an outer membrane and an inner membrane. The outer membrane turns out to look suspiciously like a eukaryotic membrane, and the inner membrane looks suspiciously like a prokaryotic membrane. I happen to see lots of folds inside of that inner membrane, what we call the cristae. And if we have lots of folds, we're increasing surface area. If we increase surface area, we're increasing whatever is going on there. So there's some function going on in that membrane. We have a space between those two membranes that we call the intermembrane space. There's that space, the cytoplasm of this thing, and we call that the matrix. We also happen to notice it has its own ribosomes, whatever those are, and it happens to have its own DNA. What we'll notice when we look inside is a cycle called the Krebs cycle. The Krebs cycle turns out to take a 2-carbon molecule, so we take the pyruvate and we rip off a carbon and we form CO2 and a 2-carbon thing called acetyl-CoA. If I take this 2-carbon and I combine it with a 4-carbon, I make a 6. And then if I rip 2 carbons off, I'm back down to a 4. So I take a 2 to make a 6. I rip 2 off, and then I get back to a 4, and I just keep the cycle going. In that process of the Krebs cycle of taking 2 carbons, take pyruvate, 3 carbons, rip off a carbon, you're left with a 2-carbon, combine it with a 4, and then remove 2 more, and you're back down to the 4. What you're doing is you're producing CO2. Actually, you ended up making 3 CO2s, which takes a 3-carbon pyruvate and turns it completely into CO2. And we make a lot of energy products. We make lots of NADHs, and we make ATP, and we also make this weird thing called FADH2. FADH2 and NADH both turn out to be energy carriers or electron carriers, which is the main thing that happens during the Krebs cycle. We produce a lot of energy, or at least energy carriers. So all that goes on, and the question then becomes, well, how does the thing know when to work or stop or anything like that? And it turns out it follows a process called enzyme feedback or feedback inhibition or a negative feedback loop, depending on whatever terminology you like to use. And here's what this turns out to be. So if I look at this picture here, I have glycolysis being turned into, in the process of glycolysis, we turn glucose into pyruvate. Along the way, we're going to produce NADH and ATP. NADH, it turns out, I can use to make ATP. Well, what happens if you have too much ATP? Do you need to keep going through glycolysis? And the answer is absolutely not. You don't need any more ATP. We have enough. So ATP is capable of binding to one of the enzymes of glycolysis, and when it binds, we change the interactions of the protein. If I change the interactions of the protein, I change the shape of the protein. If I change the shape of the protein, I change its function. And ATP has enough pull that it changes the shape of one of those enzymes, in this particular case called phosphofructokinase 1, and it's going to change its shape just enough to ensure that phosphofructokinase, that enzyme, stops functioning. Well, what does that do? It shuts down glycolysis. Well, why would you do that? If I don't go through glycolysis, I stop making ATP. But that's bad. Not unless you have too much ATP. So then what happens when you use up all your ATP? If you run out of ATP, there's no ATP to stop the enzyme, which means glycolysis starts up again. The role of feedback inhibition is it is an outside brain that can sit there and say on or off. Except I don't need a brain, I don't need nerves, I don't need cell signaling, I don't need to have a receptor transduction pathway and a response. All I need is something I end up making to come back and say, stop it. That's all we turn out to need in order to make this work. It's the most efficient way that we can have to control a pathway. But we eat more than just sugar, so how do you oxidize things like amino acids? There's 20 different types of amino acids. They all have different shapes. And it turns out the amino acids are all the words in red. We can break them into parts and put them into various parts of glycolysis and the Krebs cycle. The cycle at the bottom would be the Krebs cycle. Citrate is the 6-carbon thing. Oxaloacetate is the 4-carbon thing. Acetyl-CoA is the 2-carbon thing. So I can stick them into various parts, and I could also see where glucose all the way to pyruvate turns out to show up, which means I have different places I can stick these amino acids. The result is, if I want to burn amino acids, all I need to do is have glycolysis and Krebs cycle, and I can connect all of the dots. But what if you wanted to burn fat? How does that work? Burning fat is the exact same process. I can take my glycerol and my fatty acid chains. Glycerol is a sugar which I can stick into glycolysis. My fatty acid chains are usually even numbers, which means if I break them off 2 carbons at a time, I make acetyl-CoAs. Those 2 carbon pieces I fed into the Krebs cycle, and the result is I can just burn all of this stuff up and make a whole bunch of ATP. Seems rather easy to me. All we need to burn sugar, fat, or protein is glycolysis and the Krebs cycle, and we can do whatever we wanted. So ultimately, we end up having a lot of FADHs and NADHs, and the question then becomes, well, what do we do with all of this? Much like with photosynthesis, along that membrane, that inner mitochondrial membrane, we have a series of proteins. What they will do is they will take electrons from NADH and FADH2, and they're going to move those electrons around from complex, what we would call complex 1 to 2 to 3 to 4. Here we're not showing complex 2. It's showing 1, 3, and 4. And as they get moved, they pump protons. If you start from complex 1, you pump 3 protons. If you start at complex 2, you end up pumping 2 protons. Wop, wop, wop. The result of all of this is we have an area with a lot of protons in it, high proton concentration. It also has a lot of positive charges, so it has a charge difference. And if I allow these protons to move, why would they move? Because there's a concentration difference and a charge difference. If they move through this turnstile that we call ATP synthase, the result is it twists around. Every 3 twists produces an ATP. And what we end up making are ATP from the electrons from NADH and FADH2. The big question that then should show up is, but what do you do with the electron at the very end? That is going to be the exact same conundrum that we had with where do we get the electrons in like-dependent reactions. Where do we put these electrons? Well, let's find this toxic gas that we'll call oxygen. If I add electrons to oxygen and then I add some protons to oxygen, I can make water. And now we know why you make water during cell respiration. When you burn fat, when you burn protein, when you burn sugar, one of the byproducts is you end up making something that we call metabolic water. And now you know how camels survive in the desert.